Alaikum friends, how are you all? I am fine and hope you are all fine too. So, I am going to be teaching you the topic viruses. So, the very first question that should be asked is, what are viruses? So, a virus is an infectious agent of small size and simple composition that can multiply only in living cells of animals and plants or bacteria. So, what are viruses? Viruses are actually infectious agents. Infectious agents are those who cause diseases, and they are of small size. They have simple composition. Like they don't have any metabolic activity, or they are not super complex, and they can multiply only in living cells. Living cells means the animals, the plants, and bacteria. They can multiply only in living cells. They are totally dead outside the host. So, moving on to viruses' general characters. So, the very first character is they are ultra microscopic. They are super duper small so that they can be seen only in electron microscope. The second point is they are disease causing. But I have mentioned it intracellularly, means that they can only cause disease within the host. They are non-living outside the host. They cannot cause disease outside the host. The third point is they are composed of nucleic acid, which is either DNA or RNA surrounded by protein coat. It means that they have DNA or RNA. DNA, RNA both cannot be present in one virus. Either the DNA or RNA is present in single virus, and this nucleic acid is surrounded by protein coat. So, like this, they have DNA or RNA in their nucleic acid, and it is surrounded by a protein coat. Okay, so the next thing which I am going to be teaching you is like they have living characteristics. So moving on to their living characteristics, which is they have ability to multiply intracellularly. They have ability to multiply intracellularly. They can cause disease intracellularly, and they can not only a single virus can cause are pandemic disease. So they can multiply in living cells, and because of their multiplication, because of their duplication, their disease spread in the whole body. Okay, so the next point is they cause disease intracellularly. I have mentioned it above. Have nucleic acid, protein, and enzymes. They have nucleic acid, either DNA or RNA, and the nucleic acid is surrounded by a protein coat, and they have enzymes. What are the functions of enzymes in viruses? The function of enzymes in viruses is that they can degrade the host cell. And when the virus enter into the host cell, multiple functions are there that are followed by enzymes. Last point of their living characteristics, they undergo mutation. Okay, so the next point, topic, or the next subtopic of viruses are the non-living characteristics. So the very first non-living character is they are inability to multiply extracellularly. I have mentioned this above, that they can only multiply inside the host cell. Outside the host cell, they are completely dead. The next point is absence of metabolic activity. They have no metabolic activity. They can do nothing without the host genome. They use host genome and their host metabolic activity to do their functions of disease causing. They have no metabolic activity. Next point is absence of protoplasm. Absence of protoplasm means they have no cytoplasm, no any organelle. They just have the nucleic acid, the protein coat, and the enzymes. They have no organelles, they have no cytoplasm, and they can be crystallized. Like outside the host, they are completely dead, so they can be crystallized. Because of their crystallization, they are sometimes called non-living. Okay, so let's move on to the history of viruses. In 1660s, in Western Europe, drawings and paintings were made of tulips that demonstrated flower symptoms of viral disease. Like in Western Europe, most of the drawings and paintings were made of tulips. Tulips are flowers, very cute flowers, and in tulip flowers, like disease spread, their color changes or maybe their shape changes. So scientists must undergo through the verification of these symptoms. And in 1880s, Adolf Meyer, Dmitry Ivanovsky, and Martinez Beijerinck demonstrate TMV, tobacco mosaic virus, the very first virus which is demonstrated on earth. So let's move on to our next scientist, which is Adolf Meyer. What did Adolf Meyer do? It is disease transmitted by the sap of infected leaf to healthy plant leaf due to bacterium. He said that the disease transmission from infected leaf to healthy plant leaf, and the disease spread. So he said that the disease could be caused due to bacterium. But Ivanovsky in his research said that infection passed through bacterial filter remains infective. He passed the infection through the bacterial filter, and the infection passed through bacterial filter, and the infection remains its infectivity. So he said that it is not due to bacterium. Beijerinck said that he named the infection virion like poison, like because they pass through bacterial filter, so it cannot be bacteria, it is some other microorganism that cause disease. So he named that microorganism virion, and in other language it is called poison. The next scientist, which is W.M. Stanley, he said that infection in crystalline form, he passed the infection into the crystalline form and it remains in infectivity. उसके बाद he observed TMV. TMV क्या है? The very first visualized virus by electron microscopy and X-ray crystallography. The dated are above. And the next point is in 1915, Frederick, who was a scientist who first organized bacterial virus, who first described bacterial virus. The very first bacterial virus was described by Frederick in 1915. And because of continuous research on viruses and because of continuous verifications on this microorganism, the molecular biology introduced due to continuous study. And in 1898, Toffler and Frost, who described animal virus, who demonstrated animal virus. And the next, in 1901, the very first human virus has been described, which is yellow fever virus, and is described by Walter Reed. Thank you so much for watching my video. Hope you guys liked it, and please like this video. Thank you.